we have today a generic column of a rat built from 10,000 cells. We can now connect different columns to each other, composing 10 columns or so, simulating the electrical activity of 10 columns like this, receiving a particular input, looking at the activity, trying to understand the collective activity due to this connectivity and electrical properties of synapses and neurons. But this is from the mouse, and of course a natural question will come, so what about human? And there are several places in the world. One unique one is in Amsterdam, in the Vrije University. A group there is working in the hospital with patients, whereby the patient has a tumor or maybe has an epileptic seizure, and you have to take out a piece of a brain. So this is a brain of a human with a particular region that you have to go through, so the physician takes on the way to this region, he takes out parts of the cortex of the patient. Sometimes a big part, look at the brain of human versus the brain of a mouse. So sometimes a big part like this. So this is this group from Amsterdam. They receive from the physician a big chunk of human cortex, which is a very rare material, only comes from hospitals, living cortical human brain, and then, because this piece had to be taken out for purposes of medicine, or because of the epileptic seizure, or because of the tumor, you have to take this piece out. Then you can do classical recording. You can see a cell from a pyramidal cell from human. The cell body, the dendrites, look very similar to the mouse, bigger, with more synapses. You can reconstruct the apical tree, the basal tree. You can do really a full, complete anatomy. As you know, you can see the layers, layer 1, layer 2, 3, and so forth. You already know about the layers, and you can really characterize, like Ramon y Cajal already did, but this is now a living material. And then you can both do anatomy, reconstructing the cells, but you can also do physiology, recording the spikes from this cell, from this cell, from this cell, and also from pairs of cells, trying to see who is connected to who. And so you really can start to get a human cortical column. So we are aiming, of course, at simulating the human cortical column in the Blue Brain project. But here is some limitation. So I'm showing you the computing speed, how many operations per second these large, big supercomputers can do. We are somewhere here today. We are somewhere here. And so we can simulate about 100 cortical columns, as I said, using the present state art computers. But in order to really reconstruct, and I mean simulate mathematically, a whole human brain, we need something much more powerful, probably million times more powerful than present day's computers can do. So we cannot use present day computers to eventually simulate a whole human brain and then try using this simulation to understand the activity within the whole brain associated with a particular disease. We can simulate a column or several columns of humans, and this will be hopefully enough to already understand some diseases emerging within subnetworks. So what happens here? This is the big problem of computers today. You may know that increasing the speed by 1 million to exaflop, exascale computation relative to the petaflop or actually the teraflop we can do today, this is a big jump. This is the red jump, which is very, very difficult. And there are a lot of companies and a lot of attempts to get to this scale within 10 years. So we are here somewhere we are trying to go there in terms of simulating the whole human brain. We are hoping that due to developments in the computing world, but also due to the Human Brain Project, we will be able to develop new technologies, maybe even inspired by the brain, to be able to simulate the human brain itself. So you can see that the human brain have to generate technologies, perhaps inspired by understanding its own brain, in order to be able eventually to simulate and understand, in terms of simulation-based research, the brain of the human itself. So let's go and complete this lecture by talking about the Human Brain Project.